Hello, everyone, and welcome to today's webinar. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing this presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome Brigitte, who will speak to you more about Promega and our speakers today.
Thank you, Michelle. Hello. Also from my side, I wish you all a warm welcome to today's webinar on a fast and reliable method for SARS-CoV-2 wastewater surveillance. My name is Brigitte Saul, and I'm the commercial lead for the wastewater program at Promega. Promega is a private biotech company based in Madison, Wisconsin in the US, and we are manufacturing reagents, instrumentation, and workflow solutions for molecular biology laboratories since more than 40 years. Promega offers products for research, but also provides solutions for clinical diagnostics, forensic genetics, and applied and environmental testing. Since March 2020, we have closely followed activities in the wastewater-based epidemiology community on identifying methods to quantify SARS-CoV-2 in wastewater. We have actively participated in inter-laboratory evaluations, such as the first large one in the US initiated by the Water Research Foundation. We also received input from numerous groups who helped us by providing feedback on prototypes that we made available. We learned about the challenges of molecular testing from wastewater and about existing methods and their limitations for this specific target, as coronaviruses differ from waterborne pathogens. This is, by the way, a good opportunity to thank you all for your feedbacks and suggestions that we received over the last 18 months. So, we started by developing a sensitive and inhibitor-tolerant RT-qPCR assay specifically designed for testing wastewater samples. Now testing wastewater on a weekly routine ourselves, we also experienced the pain of lengthy concentration pre-processing methods and also realized that reliable quantification standards were missing to the community. Leveraging our expertise from other business areas, we identified components that were suitable to overcome the challenge identified and further adjusted them for the purpose. So, well, let's all warm up a little bit and maybe start with a short survey. So, for those who already do wastewater testing in the audience, please share with all of us how long it takes your laboratory for receiving the sample to get to the quantification results. So please choose one of the options that we provide here in the poll. Okay, I see it's more than the entire working day. Let me refresh. Okay, now we get some more answers. Okay, so many of you see that it's more than the entire working day. Well, thank you. With the solution that we have developed, we are now offering a workflow solution that allows laboratories to quickly implement the wastewater surveillance testing. And when we did the product development, we always kept in mind that there's a need, what the needs are for testing laboratories. And we tried to develop a workflow which is efficient, reproducible, and also includes validation features, and that they should generate consistent and reliable data well within a working day. A lot of developmental work was also dedicated to the manufacturing of standard RNAs for the qPCR assay. So we are pleased and proud to share the insights and details about our developments to support routine monitoring for SARS-CoV-2 genetic signals in wastewater. I will now introduce today's speakers, who will share their findings with us. We are very pleased about the contribution from Dr. Carlotta Olivero, who is a scientific collaborator at the Regional Environmental Protection Agency ARPA in Piedmont, northern Italy. And during the last year, her team evaluated and implemented SARS-CoV-2 testing in wastewater, and Carlotta will share their experience and results with us. Prior to her current position, she was a postdoctoral fellow at Cardiff University in the UK, where she was working on the infection of human papillomavirus in relation to the development of skin cancer. Thank you, Carlotta, for putting the presentation together and for sharing your findings with the audience. The first talk will be given by Subhanjan Mondal, my R&D colleague who leads the method and product development of the wastewater kits at Promega. Subhanjan is a senior research scientist at Promega and has been with the company since 2012. Prior to joining Promega, he was a postdoctoral fellow at Harvard Medical School working on signal transduction in innate immune response. So thank you, Subhanjan, for your great and proactive development work during that last year. And now I give you the floor. Thank you.
Thank you, Brigitta. So I will talk to you about, from a perspective of a reagent manufacturer, how we studied the subject and how we responded and developed tools to help the research community and the testing labs. So wastewater-based epidemiology is the quantitative detection of chemical and biological signatures in wastewater to analyze the health status of a population. So prior to SARS-CoV-2, the technique had been used for monitoring prevalence of polioviruses, other enteric viruses, as well as drugs of abuse. But it came to the limelight with the SARS-CoV-2 pandemic. And there has been a global effort to use this technique as a surveillance tool all over the world. So soon after the virus was detected, the SARS-CoV-2 virus was detected, it was found that the genetic signatures could be detected in the human feces. And so, thereafter, the group of Geert and Medema from the KWR in the Netherlands published a work where they were able to detect SARS-CoV-2 genetic material in the wastewater samples. And that kind of triggered the whole community to use this method, the detection of SARS-CoV-2 from wastewater, as a surveillance tool for population analysis for epidemiological purposes. And not only is the samples taken from the wastewater treatment plants, even points upstream of the wastewater treatment plants, like residential or workplace areas, hospitals, schools, or even like airports, or even more focused individual airplanes are also being analyzed. So this technique has a lot of potential as a low-cost trending tool. And there has been a global effort in implementing a more concerted effort on building up dashboards, combining them with clinical results. And it's not just a few countries, many countries. Here, examples shown from Spain, Sweden, the US CDC. They have a program called NWSS, and Netherlands. Many other countries have also, as well as smaller regional states and counties, also have their own dashboards to make the public aware of the trends of wastewater and the clinical trending. So what is being done? What is the process? What is the workflow? So once the sample is collected, the sample could be collected by composite sampling or grab sampling, and then the sample needs to be concentrated for the virus. The virus concentration is kind of important because the levels of the genetic signature in the virus is diluted, because you can imagine the level of dilution even that happens in this sample. So virus concentration is important, and then extraction of the RNA, and then followed by detection by either qPCR. Most of the virus concentration methods either rely on ultrafiltration or precipitation methods, which or electronegative membrane filtration, which have some sort of bias, and we'll talk about that, and that impacting the variability in the method. So before we go any further, we'll like to take a poll question on the methods that you may be using currently in your lab. So please go ahead and fill out one of the options that are displayed in your screen. Yeah, I see the results coming. Okay, so it's kind of evenly distributed among the three options that were provided. So I'll move on. Thank you for all the participants who took the poll. So when we started with testing many of the methods that we described, like PEG precipitation, ultrafiltration, and then we started to ask ourselves, do we even have intact viruses in the wastewater? And the answer kind of started emerging at that point. And what happens is the answer to that question is no. So there are no intact viruses in the wastewater at the time when we analyze. There is no intact virus, and the reason is, by the time it passes through the gut, the human gut, the structure of the virus is already compromised, and then it has to travel days in the sewer system. By that time, the membrane integrity is almost lost. And then so we then kind of asked ourselves, if we are not concentrating virus particles, how are we even detecting the RNA from what is it in the SARS-CoV-2 RNA that is protecting it through the passage to the wastewater system? So somehow it must be protected. Like if you spike in naked RNA in wastewater, it would be degraded in a matter of a few hours. So what is it that is protecting the RNA? So, and it happens that the nucleoprotein complexes, like say nucleocapsid proteins and other proteins that are associated with the RNA, is kind of helping the RNA and giving some protection. So then we asked ourselves if the virus concentration methods are kind of using either a size of macromolecule particles, like in precipitation methods, or which are centrifugation based, or electronegative membranes, which are charge based. So there is an inherent bias, and that could be the reason why we initially had challenges with these methods. So we wanted to develop a method that kind of addressed all these issues. And the fact that we were also experiencing challenges with these methods, others were also experiencing challenges with this method. So our thought process was, if the end goal is to extract nucleic acid for qPCR analysis by qPCR or other molecular methods, so it is best to extract the nucleic acid from the get-go, from the sample itself, from the right away sample itself. So what we came up with is a nucleic acid purification method that is more consistent. It can process less sample volume, and it can be scaled up and down. And the most significant advantage that I feel was the time saver. that you can process the entire work extraction within a few hours. So what we ended up doing is combining the virus concentration and the RNA extraction steps in one workflow. And for that, we have developed two kits. One is a manual method using spin columns, which we term as the Wizard Enviro Total Nucleic Acid Kit using spin columns. And then the next one is Maxwell RAC Total Enviro Nucleic Acid Kit, which is part manual, part automated using a Maxwell automated liquid handler. So how does this work? So the first step, here we are using 40 ml sample. That's the typical volume that we use. It's kind of a nice spot because you can use the laboratory 50 ml Falcon tubes for analysis. So the first thing we do to the sample is we add protease, add a protease solution, and that treatment is for half an hour. So this treatment is very important, and it leads to a greater recovery of RNA, here shown in the graphs by MS2, which was used as a spike control, recovery control here, as well as the SARS-CoV-2 RNA, about twofold increase if you add the protease solution. So what the protease solution does is it hydrolyzes the proteins that are associated with the nucleic acid and protecting it. So it is releasing the nucleic acid to the solution. Then what we do is we add to that solution a bunch of cheotropes and alcohol, and what that does is it kind of breaks the hydrogen bonding between nucleic acid and water and makes the nucleic acid very sticky that it can stick to matrix, like a silica matrix. And that's what we do. Then we pass that solution after adding cheotropes and alcohol, isopropanol. We pass it through silica resin by a vacuum manifold that many of you maybe have used in the past. And then the solution is the captured nucleic acid is then washed for removing inhibitors by inhibitor removal, like we use like column wash 1 and column wash 2 buffers. They are basically to remove any inhibitors that may have associated. And then the way to elute is just by adding water to it. So you are putting back the water, and then the association with the silica matrix just falls, and the nucleic acid is then eluted. So this is the first step of the concentration. Then we go through another step of concentration, and at that step, what we purified in the first step, which is like from a 40 ml sample to a 1 ml sample, that 1 ml sample is then further concentrated to say 80 microliter or 40 microliter by either a spin column as shown in the top, or by using an automated liquid handler like the Maxwell particle handler like the Maxwell RAC. And shown in the table here are some of the recoveries, the percentage recoveries for different matrix spikes that we have tested for. So for MS2, which is a bacteriophage, we've consistently got around 40% recovery. For coronaviruses, OC43 and 229E, it was around for 63% and 40% respectively. So furthermore, depending on the concentration of the virus in the sample, the sampling can be scaled up or scaled down, the volume of the sample. So what we described before was with 40 ml sample, but it can be scaled lower with lower volume. And what you need to do is to scale down the binding buffers and isopropanol accordingly. And then if you want to scale up, if the concentrations of the virus is low, then you want to scale up the volume, and then that can also be done. The graphs below show how much nucleic acid is extracted. There's a proportional increase in the amount of nucleic acid as you go higher in the sample volume. Very logical. We've also tested the workflow with sludge and suspended solids. Some countries, like in Canada, they have been using sludge and suspended solids for analysis, and that sample type kind of does associate a lot of nucleic acid. The fact being, you have to understand what the sample is. So wastewater would have urea from urine. It has bile salts, and it also has like cellulosic material, either from tissue paper or dietary cellulose. So they all could help in association to the solid. So some countries do use sludge and suspended solids as a sample type to analyze. Yeah. So now I'll talk about some of the customer feedback. So many of you have provided us with comparative results, and this kind of is important because changing a method in middle of your campaign is often a tough call. And it has to come with a big plus. It has to be taken with care. And if it is, if the method is sensitive enough that it can provide advantage of using lower sample volume or a better sensitivity in prediction, like maybe a few days of advance notice to your current method, those are the factors that kind of are critical if you are in the process already using a method and are considering or hesitant to change. These are some things that are important. So this is data from a group in Sweden, from Anna Jekli. So they had started doing their wastewater surveillance with electronegative filtration. And their method was very labor intensive, using a lot of sample. A lot of sample is not a concern because the sample volume is not a concern at all. It is the hands-on time that is a concern. But however, with a 40 ml sample, they could get way more recovery for both SARS-CoV-2 signal, here measured using the N1 gene, as well as a PMmov, which is a fecal indicator. About 20 times higher recovery than their existing method. And they changed in between, they changed their method to the direct capture method. This is another data set from a group in France, from the group of Jean-Luc Bailly. Here, what they are doing is comparing preg precipitation with the direct capture using the pure yield columns. Pure yield columns are the silica columns. So here they are testing for two SARS-CoV targets, E gene, the envelope gene, and the N2, the nucleocapsid gene. And for both, you see that the direct capture using the Maxwell method have a way higher recovery compared to the preg precipitation. And these are different samples, and for all the samples, the direct capture method was superior. So next, we will move on to the next part of the talk, which would be on the detection part, how we went about thinking of the right design for a qPCR assay, and what type of internal controls one would need to have. So as we were developing our method and starting to share prototype kits with many of you, the CDC came up with a kind of a brief outline of a kind of a guidance on how to do the RNA measurement. what type of analysis to include in such tests. So one is the assays have to be quantitative, and it makes sense if you are following trends, if you are following trends over time, which is unlike clinical testing. Clinical testing is whether a sample VTM tube that has positive or not. So that's just a yes or no type of answer. But in the context of wastewater surveillance, you need quantitative measurements. So what type of controls do you need? Then way to assess inhibitors, which absolutely makes sense. And then have some internal controls like fecal markers, human fecal markers as internal controls. And of course, measure the SARS-CoV-2 signal. So, we were kind of pleased to think that the way we were going about thinking through the analysis aspect, this was in very much in line with what CDC had put forward as well. So the specific topics that I would touch on is the quantitative standards, how we went about making the quantitative standards for the RT-qPCR assay. I'll talk about the primer probe mix. We'll talk about the GOTAC Enviro master mix, the DNA polymerase and the reverse transcriptase mix, as well as how we assess for inhibition. We call it as internal amplification control, or IAC. And then we'll also talk about the quantification of SARS-CoV-2 signal, as well as normalization by PMmov. So first, the quantitative standards, how we went about doing this. So what we did is we cloned the N and E fragments of the SARS-CoV-2 genome in a vector, PGM3Z, and this vector has a T7 promoter in it. We also cloned a fragment of the PMmov genome that we amplify in the qPCR assay, and we also cloned it in the same vector but a different vector. So then we linearized the plasmid and we in vitro transcribed to make RNA. The RNA, the in vitro transcribed sample was then treated with DNase to get rid of any DNA from the mix. And we quantify both by fluorescent dyes as well as droplet digital RT-PCR to determine the amount of RNA that would correspond to 4 million copies per microliter, and that is what we use. We dilute the RNA to 4 million copies per microliter, and that is used as the quantitative standard. So we also performed some stability study, because one of the arguments is RNAs are not stable. And these are RNAs at very low concentration. So we wanted to understand what would be the loss of activity, or if there is any, from repeated freeze-thaw. So we did some freeze-thaw experiments, and we were pleased to find that there is no significant loss of activity or degradation of the RNA by multiple freeze-thaws. Then we'll talk about a little bit more about the primer probe mix. So the primer probe mix is a multiplexed assay for measuring one of the SARS-CoV-2 targets, either N1 or N2 or E, and this would be detected in the FAM channel. It is multiplexed with detecting a PMmov, which is a fecal marker. So that would be in the Cal4 560, which is the Sci5 channel, and IAC would be in the hex channel. So the next topic is on the fecal marker PMmov and why we use that as a process control. So PMmov stands for pepper mild mottle virus. And we use that as a human fecal marker because it is prevalent throughout. It is pepper virus, plant virus that infects peppers. But it happens so that the human diet has evolved such that this virus is present in our digestive system, and it is a good marker for human fecal. As a human fecal indicator for fecal pollution. We chose this versus other established markers like crasphage and HF183 bacteroides marker because it was RNA, single-stranded RNA virus. That was the reason why we chose this over other DNA markers. The IAC stands for the amplification control, and this is in the hex channel. What its purpose is to measure the amount of inhibition, PCR inhibitors in the reaction. So if there is any PCR inhibitors in your reaction, the CT value or the CQ value of your reaction would have a right shift. So the trace below shows the increasing as you increase with the humic acid. Here, humic acid is used as a PCR inhibitor. It's a known reverse transcriptase and a Taq polymerase inhibitor. As you increase in the humic acid concentration, the CT value shifts to the right. So for no humic acid or no template control for nucleus-free water, you will have it at a lower CT value. And as you go higher in the humic acid concentration, there is a right shift in the CT value. And this difference, the delta between the CT values, is kind of indicative of how much inhibitors you would have in your sample. Typically, we say if the delta CT is more than 2, compared to the no template control, that means that you have significant inhibitors present in your sample. But it is totally up to the user how to define that. We at this point we do not provide any guidance on normalizing for that, and neither does other investigators but it is useful to know if there is inhibitors in the sample, but not to use that to normalize for anything, because there is so much other factors that could also impact. So now, once how to set up the standard curve and the RT-qPCR reaction. So you take 2 microliters of the RNA standard and for SARS-CoV-2 and the PMmov in 200 microliter reaction and do a 10-fold dilution. Then you also have a no template control, which is basically a nucleus-free water. You also prepare your reaction mix by mixing the Taq polymerase, the GoTaq master mix, the Go script, and the primer probe mix in appropriate amount, and then make up the volume to a 20 microliter reaction. Then typically very typical cycling conditions. And then these are the table below shows kind of the performance parameters for the 4 targets for N1, N2, and E, and the PMmov targets for slope, y-intercept, efficiency, and the LOD of the assays that are contained within the multiplex reaction. So at this point, we'll take a pause and take a poll. So please go ahead, answer this question. So do you implement process controls and QA/QC for the wastewater analysis? Thank you. So most of you have said that you do implement process controls and QA/QC parameters. Some of them do not, and some want to. Yes. Thank you for all the participants for this poll question. So next we'll move on to how to interpret the data. So in the assay as we described before, measures one of the SARS-CoV-2 target in the FAM channel, the fecal marker in the Sci-Fi channel, the PMmov, and the internal amplification control in the HEX channel for the inhibition control. So if you have any of the FAM channels, so we have master mixes for N1, N2, and E separately. And if any of them are positive for the FAM, we would imply that the SARS-CoV-2 signal is positive. So another thing to note there is the no template control should be negative. So if that is negative, the no template control is negative, and there is signal from the sample. That means SARS-CoV-2 is detected. Next, if the process controls all show positive signal, but there is no signal from the SARS-CoV-2 markers, that means there is no signal detected. There are two other possibilities of invalid. Could be a false positive or a false negative, where you have a signal from the NTC well, which could then basically mean you have may have a false positive, so you repeat the qPCR assay, or you have no amplification for probably pretty much anything, any of the controls as well as your SARS-CoV-2 signal. So then maybe something has gone wrong. So one case where this can happen is where you have not made up the volume of the PCR reaction properly. So say certain components in the PCR reaction is not adjusted properly. Say like, for example, magnesium, an important component for the PCR reaction. So moving on. Next, we'll talk about quantification of the analysis, how we measure viral genome copies per liter, because this will be used for the trend analysis. Copies per liter is generally accepted as the unit of measure. So what you'd need is copies in the RT-qPCR reaction divided by the volume of the nucleic acid extract that you are using in the qPCR reaction times concentration factor, and that multiplied by 1,000 for a liter. So specifically for the copies in the RT-qPCR, that can be determined based on the RT-qPCR assay and extrapolating from the standard curve. The volume of nucleic acid extract used in the RT-qPCR assay is how much you use. Could be 5 microliter of the extract from your purification method. If it is 5 microliter, then the volume in milliliters will be 0.005. So that's that value. And the concentration factor is basically how much are you concentrating. So if you are starting with, say, a 40 ml sample, and then finally you are eluting in 80 microliter in the sample, then the concentration factor is 500. If you are eluting in 40 microliter, then it is 1000. Then the next is you would also do the same thing for both SARS-CoV-2 as well as PMmov. And what we kind of have been doing in practice is doing a relative SARS-CoV-2 signal, where we divide the SARS-CoV-2 copies per liter with the PMmov signal, and that kind of helps in normalizing for the general population, as well as any dilution events, like say rainfall, or for normalizing for events like that. The PMmov signal would also be proportionally diluted, so it would is a good normalizing tool. So we have been following 3 wastewater treatment plants in the Dane County area in Wisconsin, and this is a trace from one of the wastewater treatment plants that we conducted from October of last year to January of this year. And the trace in the dotted lines is kind of the 7-day moving average of the clinical cases, and the trace in bolded lines is the normalized SARS-CoV-2 signal, and we can see that there is a good correlation between the 2 traces. The wastewater levels as well as the clinical cases. We also have published a paper on our work, which describes what we talked about in much greater detail, as well as what else can be done with the nucleic acid, like for sequencing applications, variant analysis, and following our publication, other groups have also published. The second paper is from the group of ARPA Piemonte in Italy, and you'll hear that story in much greater detail from Carlotta. And there are other papers that are also published and in press. And I will end by showing another example of how our kits have been used for implementing wastewater based epidemiology, and this one is from Sweden. Here the trace in the left is a long duration test where you can see the spike that happened with the in the March time frame was from the alpha variant. And then now it is kind of low, but in some cases that is spiking up. The trace in the on the right side, it is more zoomed in for some of the sites where there is an increase in the last few weeks. There has been an increase, and many of them are in cities or towns which are like university towns like Umeå. There is a greater increase than other places. So with that, I will pass the talk to Carlotta, who will talk about her work. Thank you.
Okay, thank you very much, Brigitte and Mondal. So I hope you hear me well. Okay, perfect. Okay, so good morning, good afternoon, everyone. I'm Carlotta Olivero, and I'm working at the Regional Center of Molecular Biology at ARPA, that is the Environmental Protection Agency of Piedmont. So first of all, we would like to thank Promega and LabRoot for the opportunity they gave us to present our experience at this webinar. And so we will present here what has been done so far in the lab about the SARS-CoV-2 surveillance in Piedmont wastewater samples. So ARPA is the Regional Environmental Protection Agency of Piedmont. It's an independent, regional independent public body with legal, technical, administrative, and financial autonomy. So the aim of the agency is to control and analyze environmental matrices like air, soil, or wastewater. So the agency is a broad and capillary collection activity throughout the region for all these environmental matrices, including the wastewater samples that have been used in this surveillance. Okay, so Piedmont is an Italian region located in the northwest of Italy. Has 4.3 million inhabitants, and in Piedmont there are 171 wastewater samples, wastewater treatment plants, and we have analyzed 13 out of them. So these 13 account for much more than half of the population living in Piedmont. So far, We have analyzed 600 samples starting from September 2020, and this monitoring is still ongoing. The samples that we analyzed were either GRAB or 24-hour composites, and all the samples of wastewater have been maintained at 4 degrees and processed usually after 24 hours upon collection. So we have followed the guidance showed in the EU recommendation number 472 that define a common approach to establish a systematic surveillance of SARS-CoV-2 and its variant on wastewater in the EU. Okay, so for the analysis, we took the wastewater samples and extract the total nucleic acid using the Wizard and viral total nucleic acid. And then we perform RT-PCR using the SARS-CoV-2 RT-qPCR kit for wastewater, amplifying the E and the N1 genes. Okay, so for setting up the protocol in the lab, we have performed a specificity test, and we ran three tests. So in the first one, we have spiked the OC43 and the 229E in the 40 ml of water. Then in the second one, other than the OC43 and the 229E, we also spiked in the SARS-CoV-2 RNA. And in the third one, we only added the SARS-CoV-2 RNA into water. So as a result, we have obtained amplification only in the solution where the SARS-CoV-2 RNA was spiked in and not in the other solution. Okay, so the data results for the monitoring have been published last August on the journal Environmental Research. For this paper, we focused on 4 main wastewater treatment plants that are highlighted in circle on the map. And the period of the monitoring was August 2020 to July 2021. Okay, so here we have plotted the CT values of SARS-CoV-2 E gene derived from the analysis performed on the 4 wastewater treatment plants on the period from August 2020 to July 2021. So as you can see, there are 2 clear and separate peaks, one in the period of October-November 2020, and the other one in March-April 2021, where the second and the third waves were in place in Italy during that time. Okay, so in order to see if there was a temporal correlation between the E gene of SARS-CoV-2 and the COVID-19 cases, we have plotted here the 2 curves together. So the blue dotted one represents the curve of E gene CT derived from the wastewater sample, and the gray one is the curve of COVID-19 cases per 100,000 inhabitants that related to the population served by that wastewater treatment plant. So as you can see, there is a temporal correlation between the presence of SARS-CoV-2 RNA in wastewater and the COVID-19 prevalence data. So the wastewater data definitely preempt and anticipate the trend of the disease for all the considered sites. Okay, so the monitoring is still ongoing, so after July, and here are plotted the data from the E and N1 SARS-CoV-2 genes for the 4 wastewater treatment plants analyzed. So again, here it's clearly visible the first peak of October-November 2020, also the one in April-March 2021. And now, these are the data from last week, so there is a tendency of a decrease in CT value, and so an increase of the presence of the SARS-CoV-2 virus in the wastewater treatment plant. So this monitoring is still ongoing, and our agency, ARPA, is sending this data to the regional health authority for further evaluation. Okay, so the monitoring is still ongoing, but we have some work in progress that we are working on. So we are planning to quantify the SARS-CoV-2 and the Papillomavirus that Subhanjan already mentioned on the wastewater samples using the standard curve method. Also, so the Papillomavirus is a fecal marker, and also that will help in the normalization of the data. Moreover, we would like to apply for the accreditation, the ISO 17025, that define the general requirements for the competence of testing and calibration laboratories. And also, last work in progress is to sequence SARS-CoV-2 and analyze its variant that populate the wastewater sample. So on this regard, we have some preliminary results that we would like to show you. Okay, so for the preparation of samples that reach and have high quality in order to be sequenced, we use the same kit that we used before for the extraction of the total nucleic acid from wastewater. So this is the Wizard Enviro total nucleic acid kit. Then we treat the sample with the DNAs, and we further perform a step of concentration and purification using the Iiac prep RNA cleanup and concentration system. Okay, so we compare two extractions. In the bottom one, you see the filtered sample. So we collect the wastewater sample, then we filter the sample twice. So first with a 0.45 micrometer filter, and then second with a 0.25 micrometer filter before adding the protease. And in the top one, we just collect the wastewater sample, and we didn't filter the sample. So we just treat with protease solution. So after the extraction, then we quantified using the standard curve method the RNA input for the library preparation. Okay, briefly, I will show you the library preparation. So after the RNA purification and QC, we perform a reverse transcription step in which we retrotranscribe the RNA into cDNA. Then we target amplification specifically for SARS-CoV-2, and then we digest the sample and run a second PCR reaction for indexing and amplification. Then we perform quality control, and then we run the sequencing. So the data then were bioinformatically analyzed using SOPHIA Genetics software. Okay, so as we expected, the output was really different between the two approaches in terms of reads aligned to the SARS-CoV-2 genome. So there were many more reads in the filtered sample, as you can see in the bottom. And whereas in the top one, the non-filter, there were less reads that were aligned to the SARS-CoV-2 genome. But the total amount of reads were nearly the same between the two samples. So as you can see, in the top, the non-filter sample, there were a total reads of 8.69%, whereas the filter one, there were a total reads around 7.93%. So we were wondering if the off-target reads were sequenced, and for doing that, we perform a metagenomic analysis. So in the metagenomic analysis, the output reads are aligned to all the genomes that are known to perform then a taxonomic classification. So as we expected in the non-filter sample, so the top one, there are 92% of the reads that are the output of the sequencing that align to bacterial genome. Whereas in the bottom one, so the filter sample, there were 61% of the reads that are aligned to the SARS-CoV-2 genome. So we plot these data on a graph and saw that if more than 40% of the reads out of the metagenomic analysis aligned to the SARS-CoV-2 genome, we observe more than 90% of genome coverage. So whereas in the case of the non-filter samples, so the blue one, there was only 30% of genome coverage because the reads that aligned to SARS-CoV-2 genome were only the 0.8%. Moreover, and most important, if the genome coverage is higher than 90% and there are more than 40% of effective reads that are the reads used to call the variant, we could define the lineage of the most abundant variant in the sample. So here we were able to define the lineage of all the samples that were filtered. So as a conclusion, based on our experience, we can say that the Promega kits we used were reliable, easy to use, and useful for our aim. And moreover, they can be used for the environmental monitoring of SARS-CoV-2 in wastewater samples, and also they can be applied for downstream high-throughput analysis. Last, I would like to thank our collaborator at the University of Turin and all the people in the ARPA agency and all the people of the molecular biology lab that run the monitoring and the sequencing. And you for your attention. Thank you.
Thank you very much, Carlotta and Subhanjan, for your presentations. We have time for some questions, and there are already many questions in the chat, so thank you for being such an active audience here. I have two questions regarding the normalization. So the first one is, do we have any idea about the PMmov distribution amongst population? There doesn't seem to be so much publication. Is there, Subhanjan, do you want to answer to that?
So the PMmov as a human fecal marker has been well studied since the 1990s, and there are many publications on the widespread. So the concept is kind of intriguing and counterintuitive, like how could a plant virus be a human fecal marker, but the way human diet has evolved over the last maybe say 100 years, the prevalence of the PMmov in human diet has been shown to be a good marker for the human fecal pollution.
Thank you, Subhanjan. There's a second question in this regard. So if, and I don't know if that was the case for you, maybe Carlotta, have you considered to use the flow rate of the water also to normalize? Have you used that, or did you go with PMmov right away?
Yes, so we use the PMmov also to our control to see, in order to see if there was dilution of the sample, maybe caused by the rain, as also Subhanjan said. So yeah, we are planning also to quantify the PMmov and to use for normalization of the data.
Yeah, thank you. I know that many groups are doing it this way as well. So thank you. There's one question, if there is an available method like a control for the extraction step. I know that both of you have done some testing with spikes while implementation. Does one of you want to comment on that? Maybe.
Yeah, I can go first. Yeah, so for the matrix spike, so the way we dealt with it is there was no consensus like what virus to use, and we have tested for comparative analysis coronaviruses, human coronaviruses OC43 and 229E, and so has Carlotta used some of the coronaviruses. But due to like challenges of culturing, maintaining, and using that as a for a starter lab, we have mostly used MS2, and for most purposes it serves the need. So but it is entirely up to the user on what best to use. Because different people are using different
validation
viruses.
then.
In the ring trials that we've taken part in, all ring trials were either with they were either spiked with a coronavirus from the organizer, the ring trial organizer, and they had also asked us to spike with any other matrix spike that we are using in the lab. So there are publications on there's some guidance on what to use.
Thank you. I see that we are up the hour now. Thank you very much. I think we have to finish this session. Thank you very much to the speakers and for everyone attending and staying with us for this 1 hour. I wish you all a great continuation of your day, and thank you for attending again. Thank you. Bye-bye.